Hello everyone, welcome to the fourth week of IMU 418 lectures. So this lecture will be about how the innate immune system is involved in both promoting cancer and can be used therapeutically to fight cancer. So as I said, the innate immune system is involved in both promoting cancer and can be targeted therapeutically. And I'll be discussing how cytokines and interferons can modulate the immune system, the innate response. And the cell types I will focus on are the natural killer cells, monocytes, dendritic cells, and MDSCs, or myeloid-derived suppressor cells. The objectives that will be covered in this lecture are to describe the different roles of the innate and adaptive immune systems in cancer and evaluate the use of innate immune cells in cancer therapy. So on Canvas, we have some articles and additional readings that are optional and a couple of chapters that cover some of these aspects in the textbook. So the innate immune cells that I'll be covering to overview, natural killer cells. So these cells are so named because they don't require prior priming by other immune cell types, and their main role in normal immune function is to kill virally infected cells and stress cells that are stressed. But in cancer, their activity and their function can be modulated in cancer immunotherapy. Myeloid-derived suppressor cells, MDSCs, these are not found in healthy people. There's not a known established role for these cells in normal function. And they are not able to be modulated by immunotherapy. They're a target for cancer therapy. Dendritic cells, they're immune sentinels. They're continuously sampling their environment to look for stressed cells or infectious pathogens. And these can be modulated for cancer therapy. And finally, monocytes, macrophages, we did cover these, more so the macrophage polarization in the previous lecture, but their usual role is in inflammatory and wound healing, detection of bacteria, pathogens, and phagocytosis. So they are both a target of cancer therapy. So those TAMs, tumor-associated macrophages, are a target for therapy, but they can also be modulated to be used as a therapeutic. So I want to go briefly over cytokines and particularly highlight a couple that are useful in therapy. So cytokines, as Dave also covered, they're small secreted proteins, and they're released by many different cell types, not just immune cell types. And they have an effect, a specific effect on interactions between different cell types, and can also be a mechanism of communication between different cell types. The cytokines, in terms of the immune system, they can be mediators of innate immunity, and the major cytokine sources in innate immune cells are macrophages and natural killer cells, but they can also regulate the adaptive immune response. But this is mainly when they're produced by T cells, T lymphocytes. So I want to specifically focus on interferons. They're one of the more well-known cytokines that are used therapeutically, and their normal function in innate immunity is to protect cells, protect from infection and inhibit cell growth. And they're a large family of multiple proteins. So there's different types. I'm only going to focus on type 1 and 2. So type 1 include interferon alpha and beta, and type 2 interferon gamma. There's also type 3, but again, I'm not going to talk about those. So each interferon signals through different receptors, but they can regulate gene transcription, a group of genes that are called the interferon stimulated genes, ISGs, and these genes are cause antiviral, anti-growth responses, and they can produce anti-tumor response as well. So this is just a picture of the structure of the type 1 interferons. You can see alpha and beta are quite similar, whereas interferon gamma it's a related structure, but it's quite different. So interestingly, interferon alpha has been approved for in the treatment of many malignancies, but interferon gamma is only approved for use in these kind of inflammatory chronic diseases, not for malignancies. But I want to start talking about these interferons and in terms of ovarian cancer, because this is related to some of the work my group does. So in ovarian cancer, it's a highly heterogeneous disease. It's got a high mortality rate because most patients will present at a late stage, and they also often recur or relapse. And at this recurrent stage or the relapse stage, there's limited options for therapeutic intervention. It's also challenging to treat because of the anatomic location, as well as the tumor microenvironment of ovarian cancer. So ovarian cancer really is mainly confined in the earlier stages to the abdomen and the peritoneum, and it does metastasize to the lung and above the diaphragm, rather than through the bloodstream. So, and there's a lot of fluid accumulation. So it's really this milieu of immunosuppressive tumor, pro-tumor cytokines, growth factors, cell types. So, systemically administered therapies won't necessarily reach the target. Especially that's why immunotherapy has been not very successful in the treatment of ovarian cancer, because the immune checkpoint inhibitors and so on, they don't quite reach the target site, and there's this hugely immunosuppressive microenvironment that they have to pass. So here's a picture of an example of the ovarian cancer tumor microenvironment. This kind of harks back to Dave's lectures on the tumor microenvironment, not just the hypoxia, the metabolic stress, necrosis, and many different cell types, and angiogenesis, lymphatic drainage, and so on. So a lot of cell types that are within this milieu that need to be targeted for therapy. And speaking of, I guess, immunotherapy for ovarian cancer, I hesitate to call this an immunotherapy because it's an antibody therapy and it's not really specific to immune cells. Is bevacizumab. So it's technically labeled as an immunotherapy, but bevacizumab is an anti-VEGF antibody, and it's not specific to immune cells, as I said. But I will discuss bevacizumab in week 6. So to talk about a novel strategy for targeting ovarian cancer using interferons. So, using the immune modulatory effects of interferons and by priming monocytes, we can deliver activated macrophage into the immune suppressive environment. So this data was from the preclinical studies of this trial, using interferons to activate monocytes for ovarian cancer treatment. And so these cells, this pointer, all right, never mind. I see. So these three different ovarian cancer cell lines, this is an in vitro study. Okay. Yeah, this is an in vitro study where the different ovarian cancer cell lines are treated with different concentrations of interferon alpha in the presence or absence of monocytes. So the black bars, there are no monocytes. This is just the effect of interferon alpha alone. And when you add monocytes, you can see in these two cell lines, the CaOV3 and OVCAR3, they're quite sensitive by the addition of monocytes. However, this cell line, the OVCAR5, it is sensitive to interferon alpha plus monocytes, but only at 20 and 200 nanograms per mil of interferon alpha. So looking at interferon gamma on its own, again, the same experiment in the presence or absence of monocytes, interferon gamma at the same concentrations as alpha. Again, CoV3, OVCAR3, these are sensitive cell lines to monocytes. Even at these low levels of interferon gamma, you get significant cell death. But OVCAR5, again, it's only at these higher concentrations of 20 and 200 nanograms for these individual interferons to stimulate the monocytes. So to look at the synergistic effect of both interferon alpha and interferon gamma together, again, the same experiment, same cell lines, these two sensitive cells, CoV3 and OVCAR3, in the presence of monocytes, even at the lowest doses, there's a huge effect. But even without monocytes, these black bars, you can see that the interferon alpha and gamma are affecting the cell line, even without monocytes. And then again, looking at OVCAR5, you can see that the effect is much, these cells have been sensitized to the activity of monocytes. When both interferon alpha and interferon gamma are in the presence of monocytes, you get a 10-fold response. So even at 2 nanograms per mil of interferon alpha plus gamma plus monocytes, you get significant killing. So this data, along with some other preclinical data, led to the approval and commencement of a clinical trial. So the trial model for this therapy is taking the monocytes, the circulating monocytes by apheresis of a patient, stimulating them with interferon alpha and gamma, and then reinfusing them to the site of the tumor, to the peritoneum, to have more profound effect. And so the patient's monocytes stimulated with interferon alpha and gamma, they signal through their different receptors to cause the upregulation of trail, which is like a depth receptor pathway. So these interferon stimulated genes are expressed on the monocytes. When the monocytes are activated, they cause cell death in the tumors through this trail and death receptor pathway, which I'm not going to go into that, but basically you're activating these monocytes and directing them back into the tumor, the site of this side of the tumor. So this approach, using interferon alpha and gamma together, will stimulate the monocytes ex vivo or outside of the patient prior to infusion, and this serves a couple of purposes. This primes the anti-tumor response of the monocytes and switches them to pro-inflammatory macrophages, and so rather than infusing a patient with interferon alpha plus gamma and then losing that effect once these monocytes might be activated, but once they get to the tumor site, then they become suppressed again. So by taking these autologous monocytes and stimulating and then introducing them to the peritoneal microenvironment, then we don't lose that important anti-tumor activity. And this is a unique approach in cancer therapy, immunotherapy, because it only uses the innate immune system. It doesn't rely on T-cell therapy or dendritic cells and so on, or antigen presentation. And it also uses the patient's own monocytes, so autologous monocytes from the patient, stimulated with these interferons to cause an anti-tumor response. So this trial is it concluded, and they're moving on to the dose escalation phase. So it's been successful in its phase 1. So now I want to move on to natural killer cells. There's a lot to cover with these cells. So their name comes from their ability to kill tumor cells or infected cells without priming by other immune cell types. So they don't require antigen presentation like a T cell does. They can kill of their own accord. They can also be activated by cytokines, particularly IL-15, can activate the killing response of natural killer cells, and they, in place of antigen presentation or activation, they can recognize cell surface proteins on normal cells and infected cells or tumor cells, and the lack of distinguishing self proteins, so these markers in, across species, they're called MHC, so major histocompatibility complex proteins. But when we're talking about specifically about humans, you'll see HLA being described, so human leukocyte antigen. And by the combination of the activating receptors on NK cells through a variety of ways, which I'll get onto in the next slide. You need both, natural killer cells respond to both activating and inhibitory signals to kill. So in this image, when you have both an activating receptor in blue and an activating ligand on the cell, that would be enough to kill, would cause the natural killer cell to kill. But because the normal cell is presenting a self marker, they're escaping death through this inhibitory receptor. This is a very simplistic model of how this works, but it'll get you by. And in this case, the tumor cell has lost its MHC or HLA marker to inhibit the killing, so you get activation of killing by the natural killer cell. So to go through that in a bit more detail, there are activating receptors on natural killer cells. And there are quite a lot, but I only want you to focus on this NKG2D, natural killer group 2D. Not a very imaginative name, but it is important because it recognizes the stress proteins, or it recognizes the activating antigen ligands such as mica. And there are, as I said, multiple others. Some specifically recognize viral proteins that are expressed. And CD16 is an important one as well. It works in a different way because it recognizes antibodies that cover a cell by recognizing the Fc portion of the antibody rather than the antigen binding part of the antibody for this very specific type of cell death called antibody-dependent cellular cytotoxicity, which, I will cover in this lecture, but it will be important for the week 6 lecture as well. So to illustrate that, activation in the absence of any inhibitory signaling, this would be a cell death message for the natural killer cell. And then for the, there are inhibitory receptors expressed on natural killer cells to make sure they don't kill self or normal cells. And this, again, it's confusing, and I would have thought of a better name, I think, but NKG2A is the inhibitory receptor expressed on natural killer cells, also known as CD94, and this is the important receptor that recognizes that self-antigen, the HLA or MHC. So expression of this HLA-MHC molecule by the tumor cell or normal cell prevents cell death by the natural killer cell by binding to this natural killer cell receptor. So as I said, alluded to, CD16 works slightly differently. So CD16 is a receptor for antibodies. So an antibody might be specific against tumor antigen, and the antibody will bind by its variable chain, but the CD16 receptor, it binds and recognizes this Fc portion of the antibody, and this is an activating response. So more about natural killer cells, as well as their cell death, they can also secrete cytokines like interferon gamma and TNF alpha, so tumor necrosis factor alpha, to activate other immune cells and recruit them to enhance the immune response. But their major mechanisms of killing, there are two types. Granulocyte apoptosis, mediated by the release of these cytotoxic granules, and ADCC, antibody-dependent cell-mediated cytotoxicity. So the granulocyte apoptosis it's really cool. We can watch this video. Basically, once the NK cell is engaged, it's activated. It's not got an inhibitory signal. It can release its cytotoxic granules into the target cell. So first there's perforin, which basically perforates the cell membrane, punches holes in it, and then granzyme and different serine proteases can be delivered into the cytoplasm of the target cell to just really induce apoptosis. And so in this first 20-second clip, the perforin is labeled in a fluorescent antibody, fluorescent dye. So you'll see how quickly and how efficiently these cells can kill. So, there's a little natural killer cell, and yeah, so fast that it injects its, granules, and it causes very efficiently apoptosis in these tumor cells. And here's just like a light microscope version of this, an infected cell. This one I like because it looks like a big implosion of a dying star or something like that. But again, the same very efficient, very fast mechanism of killing. Okay, yeah, that just continues. So the other mechanism is by ADCC. So again, this is facilitated by the receptor on the natural killer cell, the CD16 receptor, being able to bind to an antibody. So an antibody must be involved. It must label a target cell, and the Fc portion of the antibody is where CD16 binds. So, and that way it can also cause, again, this granulocyte apoptosis, or by this particular cell death receptor signaling pathway, again, trail, but I'm not going to go into that. So we can use natural killer cells to augment anti-tumor response in a couple of different ways. So the first is using an IL-15 superagonist. There are a couple of different ones that are available, but I guess the most prevalent one is this ALT-803. So IL-15 can activate natural killer cells. So this IL-15 was engineered into this agonist as a mutant. So this particular mutation of IL-15 causes it to be basically hyperactivating. And by binding it to this Fc and IL-15 receptor, you get more stability of this agonist to basically switch on these natural killer cells forever. In this panel B, cytokines, again, IL-15, and as well as others, but mainly IL-15, can activate natural killer cells. We would mainly use this in combination with therapy to get better response. These bikes and trikes are interesting strategies. So they again use the activating receptor CD16. So they make a single chain variable fragment, which is the variable fragment region of an antibody to bind to the CD16 and activate it. But they also add in IL-15 to activate the NK cells, so they have like a bifunctional effect, so double activation through two receptors, because they're bind and they won't come off, or they won't switch off. And you can also add in a tumor antigen. So this would be if you didn't have an effective antibody for your tumor antigen of interest, and you would add in your antigen. This is basically like an antibody without being an antibody. So you still get antigen binding and activation through CD16 for this ADCC, but you just enhance that without using an antibody. If for some reason your antibody doesn't bind or doesn't, or it gets internalized by the cell or degraded, this would be a strategy that you could use. These further strategies these are less, I guess, less developed strategies. This NKCES, which I missed opportunity to call them Nikes if you've got bikes and trikes, but these are essentially the same as the bikes and trikes. They activate two activating receptors. So again, CD16 for ADCC, and one of these other receptors that I didn't talk about, one of the antiviral receptors, by fusing the Fc portion of an antibody, that's what CD16 recognizes, to the ligand for this receptor to an tumor antigen, you get activation through both of these receptors. So it's a really strong activating signal. Immune checkpoint inhibitors, these are important. They're becoming, they're the main immunotherapy module. So you've probably heard of anti-PD-1, and I will cover immune checkpoint inhibitors in the antibody lecture. But basically, if you want to block the inhibitory signals, so remembering that NKG2A recognizes the MHC, the self, so removing any inhibitory signaling on NK cells in combination with activating NK cell signaling, then that would enhance an anti-tumor response. And finally, natural killer cell CAR, or chimeric antigen receptor constructs. These are less well established, and yeah, I'm not going to go into them. Basically, this involves genetically modifying natural killer cells, deriving them from a patient, genetically modifying them to cause an activating receptor bound to a tumor antigen. Yeah, as I said, I'm not going to go into these, but that's another strategy, just basically activating, causing an activation signal in response to a tumor antigen. Again, if you didn't have a good antibody that could cause this, you would perhaps consider making a chimeric antigen receptor for the NK cells. I'm going to move on to dendritic cells, and I'm not going to spend a lot of time on this because Dave talks about dendritic cell therapy in his the final lecture in week 7. But dendritic cells, they're immune sentinels, and they really are only activated in response to antigen. Once they pick up an antigen, they will migrate to the lymph nodes to present their antigen to T cells to activate T cells. The immature DC is, more polarized towards tolerance and sampling of the environment to find antigen, but a mature DC, they can stimulate T cells, and they're the activated anti-tumor response. So within the tumor microenvironment, dendritic cells are sometimes present, so they can migrate to the site. They're sampling, they're trying to uptake antigen, but they're suppressed by the tumor microenvironment, or they're just maintained as these immature DCs, so they don't migrate back out to to present antigen to T cells. So in targeting dendritic cells, there are a couple of ways to be able to use dendritic cells to be anti-tumor. You can block these inhibitory motifs. Again, it's similar to the natural killer cells by blocking an inhibitory signal. The DCs can then be allowed to mature and present antigen. You can also feed antigen to dendritic cells if they can't access it in the tumor. There's a couple of more prevalent receptors that are targeted to feed antigen to, including DEC205 and CLEC9A, but I'm not going to discuss these. And of course, DC vaccines. So it's, similar to targeting these receptors with antigen, you can essentially harvest dendritic cells, modify them by feeding them tumor antigens, and then presenting them to T cells. So this creates like a dendritic cell vaccine, which again, Dave will cover. So finally, on to myeloid-derived suppressor cells. These are the real bad ones. They have many suppressive functions and are really only pro-tumor. So there are no MDSCs that have been found or in healthy individuals. So they are a really nebulous cell type. They're quite heterogeneous. Some cells within this group will be more like neutrophils, and some will look more like monocytes, but they're a combination of neither, or they're a combination of both. And they're really only found in patients with chronic inflammation or malignancies. So as I said, they're quite heterogeneous, but they all share the ability to suppress T cell activation, so they can suppress the adaptive immune response and also promote other aspects of tumor growth, including angiogenesis. They can support tumor invasion and metastasis. So it's also been shown that the more MDSCs that a patient has a poorer prognosis on the outcome of cancer. So there are multiple ways how MDSCs suppress T cells. Specifically, they can produce a lot of factors that interfere with T cell activation and proliferation. So they can produce reactive oxygen species and nitrites that interfere with T cell receptors. Production of L-arginine prevents T cells from proliferating. And then there are other like non-specific factors that can just interfere with T cell function, including TGF beta and IL-10. IL-10 is a anti-inflammatory and it won't recruit. It is a suppressive cytokine to stop recruitment of other immune cell types. So it also can, these MDSCs can also kind of activate or increase the influence of T regulatory cells. So these Tregs are in their normal immune function is immunosuppressive, and they are involved in the generation or production or development of immune tolerance. And by secreting special cytokines, it increases the amount of Tregs. You've got an imbalance of these Tregs, T helper cells that promote suppression of other T cells, and direct suppression of the cytotoxic killing T cells. So there are ways to target MDSCs to try and help the pro-inflammatory signaling to come back and destroy tumors. So these include depleting the MDSCs by inhibiting them or killing them, blocking their migration to the tumor. You can inhibit their activity so they can still exist, but they can be devoid of their suppressive function, or also by trying to differentiate these basically undifferentiated nebulous cell types into a functional, helpful cell type, such as a dendritic cell or a macrophage. So to go through some of these strategies to deplete MDSCs, there's not really any specific therapies so far. Chemotherapy can deplete these cell types, but there is some early research suggesting that these special agonists to activate the liver receptor can specifically kill MDSCs, but this is in early phase trials. To block the recruitment of MDSCs to the tumor site, there are multiple different inhibitors that are directed against the chemokines that cause the MDSCs to be recruited, as well as STAT3 signaling can be inhibited to prevent the MDSCs from migrating and being recruited as well. To inhibit the immunosuppressive function of MDSCs, there are a couple of inhibitors. So phosphodiesterase inhibitors, PDE-S. They target those the T cell immunosuppressive factors such as ARG1 and the INOS, or a more targeted way is to inhibit COX-2. COX-2 is upstream of the PG2, which causes the ARG1 expression. So these both kind of target those suppressive molecules and functions of the MDSCs. And then finally, to attempt to differentiate MDSCs into mature anti-tumor cells. This is, I guess, the least advanced mechanism, but there are a couple of different inhibitors and metabolic proteins that can cause differentiation, though this isn't really specific. And there's also been some, I guess, early research into using HDAC inhibitors to cause reprogramming at the epigenetic level, retargeting the genome of a MDSC to try and push it towards the expression of genes to push it towards a certain phenotype. So to summarize this lecture, the innate immune system, it's not just the adaptive immune system that's involved in cancer progression or can be used in therapy. The innate immune system has a strong role and is a subject of further research for its use in therapy. So cytokines and interferons in particular They can modulate the immune activity of multiple cell types, and again, not just the adaptive immune response. They're very important in the innate immune response. And again, innate immune response, we can start looking into this type of response for targeting and augmenting this activity as a anti-cancer therapy. And the MDSCs are highly suppressive immune cells. They cause suppression and by multiple mechanisms, but also promote angiogenesis and so on. And these are a good target to try and eliminate to enhance anti-tumor responses. So for this week, there is no weekly assessment that's just based on this lecture. It will be the midterm exam. So this covers content from the lectures from week 1 to 4 and is worth 30% of the total grade. And it's a mix of multiple choice and short answer questions. So please email us if you have any problems, and I'll see you next time.